sorry, I just started recording. As we talked about before, this increases with the age of the mom, because you can think about our eggs have been sitting in prophase 1, and those chromosomes are sitting in their tetrad entangled for years, decades, before they're asked to separate. So as we age, the incidence of those not separating is great. And if you think about this, look at this, like between 35 and 40, how that goes up, how many infants per 1,000 births this goes up, and then it keeps going up. This is just Down syndrome. So if you were to graph this similarly with miscarriages, you would have that same increase in miscarriages, because it was probably happening equally on other chromosomes. It's not like chromosome 21 is somehow targeted for trisomy. That's not the case. It's that every chromosome has a random chance of being in trisomy, and this just shows you the rate of how many births come out from trisomy 21. Okay, any questions on that? Also, there can be the wrong number of sex chromosomes that can result. These are not typically in the same kind of category as the other three, 21, 19, and 13. These people typically don't have any sort of intellectual delays or developmental delays. Mostly what they have are secondary sexual characteristics that aren't quite fully formed. So sometimes they're kind of mid-formed or overly formed, so they might have really long appendages or be really hairy or something like that, that are a result of having the wrong number of sex chromosomes. Okay, so the last thing in this chapter talks about how oftentimes we get changes in our chromosome that can also result in birth defects. So you can have deletions, duplications, inversions, or translocations. All right, so a deletion would be, for whatever reason, maybe a piece of the chromosome loops out during replication for some odd reason, or maybe when you're crossing over during meiosis, it gets crossed and some part doesn't get equally put back, and so you get a part missing. Sometimes we end up with duplication. So a part of a chromosome, for whatever reason, ends up being duplicated. In many cases, duplication is a good thing. Duplicated genes in our genome are all over the place, and they evolve at an independent rate from the original gene. So sometimes the original gene might not have any mutations in it, but we accrue mutations in the duplicated copy that ends up having some new function that environment selects for, and we get some new function over it. So gene duplication has actually led to a lot of our evolutionary development over the years. You can also have a part where maybe the chromosome gets broken, gets put back together, but not in the right orientation, so it gets put back together backwards, and that can be really problematic because of gene regulation and things like that. The last one is a translocation, where you have a piece of a chromosome that gets broken, and instead of being partnered with its homologous pair, it gets partnered with some other chromosome that's not a homologous pair. We saw this when we talked about Oliver Casey and his CML. If you remember, there was a translocation between his chromosome 9 and his chromosome 22 that resulted in an activation of a kinase in a new way, and that caused him to have basically tell the cell to divide way more often than it was supposed to, and so that caused him to have cancer. So if you guys remember that, so that was a chromosomal translocation. Oh, I want to say one more thing. Sometimes trisomy 21, I'm sorry, Down syndrome is also caused by a translocation. So sometimes it's the result of inheriting too many chromosome 21s. Other times it's actually a result of part of chromosome 21 being duplicated or translocated somewhere else on another chromosome, and basically you get the effect of having 3 chromosome 21s, but really you just made some other chromosome longer with part of chromosome 21. So that's another way that Down syndrome is out there. Okay, so that's the end of chapter 10. Do you guys have any questions on 10 before we start on to genetics? No questions? All right, then we're going to get into chapter 11. This is a long chapter, and it is one of my favorite chapters. In the past, I've taught genetics at TCC, and I love teaching genetics, but we never have enough students that take it. It's not really transferable because it doesn't have a lab with it. So most universities, if they want you to take genetics, they want you to take a lab with it. But it's super, super fun to take. So if you ever see it pop up on our schedule, take it. It's just one of those that you just take for fun and not necessarily for your degree plan, because it's just so interesting. And I wish we could have a whole semester to cover all of this stuff that we have to cover today, but we're going to at least hit the highlights so that you're aware of it, and when you get to it somewhere else down the line, you'll be able to say, oh, I heard of that before. Okay, so these are your learning objectives. So we're going to start with really simple genetics just to kind of get us all on the same page, and then we're going to really branch out into some of the more difficult stuff. So just be prepared for that. Okay, so obviously that's my boys a long time ago. Genetics are how traits are passed from one generation to the next, right? So you can see that Jackson looks a whole bunch like me, and actually as he ages, he looks more and more and more like me. John Keith doesn't look like me at all, right? And I want you, as we're going through this chapter, I want you to think way big picture, right? Everything we're talking about is driven by the central dogma, right? So how we get our traits is through proteins, and how we get proteins is through the central dogma. So then you have to realize that mutations in the DNA are going to influence our proteins, which then influence our traits. And then I want you to think about it on the other side, how those differences in traits influence evolution and natural selection. So this is how the central dogma kind of links all of this together, that it's not, these are not separate parts of biology that aren't related. They all are completely intimately related with each other. So don't lose sight of central dogma as we're going through all of this. Also, don't lose sight of mitosis and meiosis, because they are what are the mechanism that's used for passing these traits to the next generation. Okay, so I'm going to do this really quickly because I love the history, and I think this is a fun one to talk about. So there have been some ideas to help explain inheritance for many, many years before Mendel came around. And some of the ideas were kind of fun. So one of the ideas that came about by Hippocrates, so in 400 BC, was that they were, and he called them pangenes, so probably where we got the word gene from. He said that there were these little parts of us all over our bodies, like maybe dark hair on our legs and a funky looking toenail or bow legs or something like that, right? And he said that at the moment of conception, all of those parts would kind of travel to the egg and the sperm and deposit themselves in the egg and the sperm, and that's how the traits would get passed on. And if you think about the idea of sexual intercourse with humans, how that might be how you're thinking, right? Because there's a feeling, a rush of a feeling when you're having an orgasm that kind of makes your whole body go like that. And so he was thinking that must be all of these little traits kind of traveling up through your body to get to the egg and the sperm, and the ones that make it are the ones that actually get transferred to the next generation, which I think is kind of just an interesting way to think about, you know, what they were thinking way back then. Another idea comes from the idea that, think about what a baby looks like. When a baby comes out, like John Keith was a preemie, he was super little. I mean, literally fit in the palm of my hand. He was so little, and he was perfect. He had this head with 2 eyes and 2 ears and this cute little nose, and he had 2 lips, and he had 2 arms and 10 fingers and 10 toes, right? He was this perfect little human, but he was just this miniature thing, right? He was just tiny, teeny, tiny, and then he got bigger and bigger and bigger. He's still miniature. He's still not as big as an adult. So people at the time were thinking, well, if babies come out looking just like mini humans, maybe there's just an even smaller mini human in the egg or the sperm. All right, so these were called, people that thought it was in the egg, they were called ovists. They just thought this little teeny tiny person, he was even smaller, you know, microscopic inside the egg. And then there were people who thought it was microscopic inside the sperm. So that's kind of a fun thing to think about. Then there was the idea of blending, that maybe if you mated, then if you had a black colored bunny and you had a white colored bunny and you mated them, you got a gray colored bunny, right? But then you would think that everybody would end up being gray over time, right? And we don't see that to be true. So then it came down to Mendel to show the particulate hypothesis. So Gregor Mendel was doing his work at the same time Darwin was doing his work in the late 1800s. And he was just kind of a curious guy. He liked science, he liked plants, and he liked math. And he kind of thought it was cool to use math to explain natural phenomenon. So what he started doing was he had a garden and he started noticing that there were these different plants growing in his garden, and so he started to question what that was and tried to control it. So he used pea plants kind of as his model system to study how traits were transmitted from one generation to the next. And pea plants ended up being like the perfect thing he could have picked. Had he chosen something else, he probably wouldn't be considered the father of genetics, but he chose something that worked very well to explain the basics of it. Because I'll tell you that what he figured out is very basic, and we know so much more that don't follow his rules now. But what he figured out was very basic. So why were pea plants so good? First of all, he could control the matings. So we'll talk about this in just a second, but plants can mate with themselves or they can mate with other plants, and he could control that. So he could either make them self-fertilize or make them cross-fertilize, which means we mate them with someone else. So we're going to use the word cross, and cross just means mating with something else, okay? And then the other thing that was really nice was the outcome. The traits that he was looking at were very distinctive, and they came in kind of two forms. So for example, we're going to look at flower color. The flowers were either purple or they were white. There was no other like teal or pink or anything like that. They were just these purple or white. And these peas, they were either yellow or they were green. And there was no orange or brown. They were yellow or green. That was it. So that made it really easy for him to keep track of numbers. He could grow what are called true breeding strains, where if he mated the plant with itself, the outcome was always the same. So they breeded true to what they were supposed to be. They didn't take very long to fertilize. If you've ever planted a pea and grew it, within days you have a little sprout coming up out of the ground. And he could plant all of these peas and get lots of offspring. So he had large numbers, so his sample sizes were fairly large, which, you know, when you do an experiment, if your sample size is large, that gives you more confidence in the data that you're collecting. All right, so a little bit about flowering plants. Flowering plants have, in their flowers, they have their reproductive organs. The petals are just basically protective structures around them. And then inside the petals, you have basically kind of like a testes and an ovary, in a sense, because those are the areas of the plant that are capable of doing meiosis. So this bowling pin looking like thing here, this is basically the ovary of the plant. It's called the carpel. And inside there are cells that go through meiosis and give rise to eggs. So there's a lot of eggs kind of sitting in this ovule part right here, okay? And then these stringy parts here, these are the cells that are going to do meiosis and give rise to sperm, and then the sperm are here at the end. This is called the anther. This whole thing's called the stamen. This is called the anther, and this is basically what we call pollen. Those are just plant sperm, right? And so what can happen is the pollen can blow off of the plant, and just think about a plant that you've had on your counter. Over time, you start seeing the pollen just kind of fall off. That eventually happens. Well, if wind blows, it can blow it and it can land right here on the top of the carpel, and there's basically a little sperm tunnel that the sperm can swim down here, find an egg, and fertilize it. All right, so that's how a plant would fertilize itself. Okay, that would be an example of self-fertilization. If it's a cross-fertilization, the pollen and the egg are derived from different plants. And how Mendel controlled that was he would basically go in and he would cut the anthers off from one plant, and then he would use a paintbrush from a neighboring plant and basically pick up the pollen grains from the other plant and then dab it onto the ovule, and they would go down the little sperm tube and fertilize the egg. Okay. This is considered a hybridization because you're mating or crossing two different individuals that have distinct characteristics. For example, one has a purple flower and the other one has a white flower. So think about a hybrid car would be the one mixing one that does gas and one that does electric. He would then take the peas that grew off the plant that he fertilized. He would plant them and grow them into offspring. Okay, and those offspring would be the hybrids, right? So when he did his crosses, the first cross he started with, he called the parental strand. Typically, these colored flowers he started with were true breeding, which means if he left this purple flowering plant to breed with itself, it would always make purple flowers. If he left this white flowering plant to mate with itself, it would make always white flowers. That's what true breeding means, all right? And then he would look at the first generation of offspring, which he called the F1 generation. Now, he didn't just do this with flowers. We're going to mainly look at it with flowers, and a little bit we're going to look at seeds. But he looked at a whole bunch of different traits that he could find that differed in these plants. So he could look at whether the plant was tall or dwarfed. He could look at whether they were purple or white. He could look at where the flowers came off of the stem. Were they axial or were they terminal? He looked at seed color. He'd look at seed shape. So sometimes they were round, sometimes they were wrinkled. He would look at pod color. So sometimes the pods were green, sometimes the pods were yellow. And then he would look at pod shape. Sometimes they were smooth, sometimes they were constricted. Every time it was one or the other. And when he studied these initially, he would hold everything constant except for one trait. So he would, for example, in that last example we did, maybe all the plants were tall with axial flowers and yellow seeds and round seeds and green pods and smooth pods. And the only thing that was different between them were the flower color. So he was careful to make sure that they were only different in one trait at a time. All right, so here's how he started. He started with these true breeding flowers. So if he self-mated this purple flower, it always gave what color flowers? I'm asking you guys. So if it's true breeding, that's right, he would always give purple. And then the same thing with these white flowers that were true breeding. So that if he made them with themselves, they would always give white flowers. And he would mate them to each other. And what he found in the first generation, over and over and over again, with every single one of those traits I showed you in the last picture, that he found that the first generation always looked like one of the two parents. Every single plant, 100% of them always look like one of the two plants. So for in this example, purple true breeding times white true breeding always gave rise to offspring that were purple. So he had to figure out what happened, right? First of all, he was disproving blending, right? Because it wasn't some, light purple or lavender color. It was the same exact color as the original purple parent. And so he started to think, okay, what's going on? It's not blending. What happened to the white trait? Is it lost for good? Am I never going to get it back? So that triggered him to go through the next round of mating. So what he did was he would mate the F1 generation plants either with themselves or with each other. So those purple flowers from that first generation, they would mate with each other. And when he did that and gave rise to the second generation of plants, he would find that the white trait would come back. All right. So, and it came back at a very consistent ratio. It always came back about 25% of the plants that were grown were white, and the other 75% were purple. And again, he did this over and over and over with all of those different traits and always got this same 3-to-1 outcome in terms of what the traits look like. 3 of them always look like that first generation color or size or shape or whatever, and then 25% of them look like the trait that seemed to have disappeared. So he had to ask himself, why did one disappear in the F1 generation, and then what made it reappear in the second generation? So this is just an example set of data, but he did this with flowers and flower position and seeds and pod shapes and length, height, and he found that across the board, he always got that same 3-to-1 ratio. All right, so Mendel then postulated from these findings the following things. Keep in mind, he did not know that DNA was a genetic material. We hadn't understood mitosis or meiosis yet. We didn't understand anything about central dogma yet. So this was all about maybe like 70 years before we knew all of that. So he was kind of really ahead of the game here. So he predicted that the pea plants had 2 discrete hereditary factors that you would get one from each parent. So we call those genes, right? You get a chromosome from mom that has genes on it, you get a chromosome from dad that has matching genes on it, and you get one from each parent when you're conceived. So he predicted that. He also predicted that those 2 factors, which we now call genes, can be identical or different, right? If they're different, then one of them seemed to dominate over the other one, right? So one of them he called a dominant trait over the other one, which was recessive and was kind of being masked, right? And he also predicted that during gamete formation, that these paired factors segregated from each other, and so the gametes only got half, and so that those 2 halves came back together. So his work really led to the thinking behind meiosis, and he was really right on. So this led to the idea of segregation of traits during meiosis, right? So they segregate during meiosis 1, really, right? When the mom chromosome separates from the dad chromosome. Okay, a couple of vocabulary words we need to make sure we know. We've talked about what homologous chromosomes mean. We know that they're the same length, the same centromere position, the genes located in the same positions. I think we talked about this word a little bit, but now we're going to talk about it way more often. When we have different versions of the same gene, those are called alleles. So when I say different versions of the same gene, what does that mean? How can you have a different version of the same gene? What does that actually mean in a molecular way?
Like they're at the same position on the gene, but they could be coded for different expressions like blue eyes or like brown eyes.
Okay, good. So they're coding for a different trait, and like that's a different protein. How do we code for a different protein? What would actually be different between the purple flower allele?
Introns and extrons.
Okay, maybe it's alternative splicing, could be. Chances are, in this case, it's the DNA sequence, right? So it's actually a change in the DNA sequence. Well, let's go here, and I'll go back to that picture in a second. So alleles are just a difference in the DNA sequence between the 2 genes that are being compared. So this one has a C right here, this one has an A right here, right? This typically, when we look at these differences, we find that they lead to different proteins, right? So the dominant allele, in most cases, this is typically true, not always true. The dominant allele codes for a functional protein. So in the case of the flower color, this dominant allele codes for a pigment that makes the flower look purple. The recessive allele codes for either non-functional protein or a reduced function, or maybe different function. So now that in this case, it's a non-functional protein. So now you have no pigment, which means you're going to be the absence of color. Okay? So if you are homozygous, that means you get 2 of the same allele, right? So if you're making brown pigment and you're making brown pigment, you're going to have brown eyes. If you're heterozygous, you're going to have 2 different alleles, brown eyes versus blue eyes. But because you're making the protein from the brown allele, you're still going to have the color that's associated with that brown allele, and you're going to have brown eyes. Same with the purple flower. If you're heterozygous, like in this condition right here, even though you don't make any enzyme from the recessive allele, you're making the enzyme from the dominant allele, so you end up still with the right amount of pigment to make you look purple. Okay? Yes, now eye color is a little bit more tricky than that because there are multiple genes involved, but in a general sense, yes, you can think of blue eyes as being the absence of brown pigment, and that's why you have that. Okay, so when we're talking about genetics, we often refer to the word genotype, And basically, when you're talking about the genotype of the cell, you're looking at one trait, typically at a time, maybe two, and you're basically indicating the allelic combination you have in that particular cell. So remember, a somatic cell is always going to have two alleles for every gene, right? You're going to have one from mom, one from dad. If you're a gamete, you're just going to have one allele, right? Because we've separated the chromosomes out. So the genotype for a somatic cell can be described in the following ways. We can talk about it being homozygous for the dominant allele, meaning two of the dominant allele. All right, so homo meaning the same allele. In this case, it's the dominant one. And remember, the dominant one in most typical cases makes the functional protein. Or you can be homozygous for the recessive allele. Again, homozygous meaning the same, but the allele you have now is the recessive one. So in this case, no functional protein gets made, so that's why you're white. Or you could be heterozygous and have one of the dominant allele and one of the recessive allele. All right, so if we go back to this picture here, what do you think the genotype of this true breeding purple flower is?
Dominant.
Okay, it's going to have at least one dominant allele. Does it have one or does it have two? It's true breeding. It always makes purple flowers.
Two.
It's got to have both, right? It's got to be big R, big R. Whereas this white one is going to be what? Homozygous recessive, little p, or little r, right? And then this one that seemed to give rise to both, purple and white, what do you think its genotype is? That's right, it's going to be heterozygous. All right, so those true breeding ones that he mentioned earlier were actually just homozygous dominant or homozygous recessive. And keep in mind, we don't just inherit one gene at a time, we inherit chromosomes. So we inherit this whole combination of allelic combinations, and it can differ from gene to gene. So if this top chromosome was from mom and this bottom chromosome was from dad, right, for gene G, we could be homozygous dominant. For gene A, we could be homozygous recessive, and for gene B, we could be heterozygous, right? So all along the same chromosome, we can have all of these different possible combinations. Okay, the other word we have to talk about is phenotype. And so the way to remember that is it starts with a PH, so that tells us a lot about the physical characteristics that we see. It's not always just physical, so a phenotype can be whether there's enzyme function or not, right? And that might not be something you see outwardly. But in many of his experiments, he was looking at physical traits like purple flowers or tall or were the seeds green. When it comes to a phenotype question, your answer is typically an adjective, right? Purple, tall, green, yellow, wrinkled, whatever, okay? It describes what it looks like. And phenotype is influenced, obviously, by genotype, but we're going to find out a little later there's also other genes that can interact with this and the environment that can interact with this. You can also imagine that, like, chromatin structure can influence this. You don't have to necessarily have a particular genotype, but if your chromatin structure suggests you don't express that gene, it'll look like you don't have that particular genotype. So there's a lot of stuff that goes into influencing phenotype besides genotype. However, for right now, we're going to kind of push that other stuff to the side and just focus on what if genotype directly influenced phenotype, could we predict outcome? All right, so couple of vocabulary things here. Typically when we talk about the normal, and lots of times in genetics we use the word normal, and it doesn't really mean that it's better than, because it might not be better than, but it's what you see most prevalently out in the wild. So in real genetics labs, they call normal the wild type, the type that's most prevalent in the wild. And then they call the one that has the mutation the mutant. And mutant sounds like it's bad, but think like X-Men or something, right? Because those are like cool mutants that have like cool outcomes from their mutations. So not all mutations are bad. Keep that in mind. All right. All right, so here's an example. This is albinism. So albinism is pretty cool. It is caused from the lack of depositing of melanin, the pigment that makes your skin color, into your skin cells. So what's cool about albinism is that the cells actually make melanin. All right, so this big cell right here that looks kind of like a blobby amoeba kind of thing, it's called a melanocyte. And its job is to just pump out melanin hugely, right? And then it delivers its melanin to the neighboring skin cells that are above it. So if you think of this as kind of dermis, like deep in the tissue, and then this is the cells on top of your skin that we see, all right? So its job is to deposit the melanin pigment into those cells. And then once you have the pigment in there, just like the flower pigment, it's going to give color to those cells. In a person with albinism, this depositing feature doesn't work right. So you can see here, these guys aren't doing a good job of depositing. And so these upper cells here don't have the coloration that they're supposed to have. All right. So the issue is with depositing. So when you have albinism, which is a recessive condition, you have to inherit both recessive alleles, and that will determine basically that none of the pigment can be deposited into your skin cells, and therefore you're not going to have color in your skin or your hair or your eyes. Okay, so in an individual who is homozygous dominant, they make and deliver the pigments appropriately. They're going to have normal wild type skin pigmentation. They're going to have the right color. Okay, if you're heterozygous, you're still going to make some of that pigment. You're still going to deposit it, so you're still going to have that normal pigmentation, right? However, if you're homozygous recessive, you're not going to be depositing any of that pigment, and you're going to have very, very white skin. Ivan.
What about those who have, blotches of like white skin and then blotches of skin with melanin?
That is called mosaicism, and that's actually like a mutation, or typically it is a mutation in just like a patch of the skin. So the cells that's giving rise to that patch of skin has the mutation, and so those cells aren't making the color, whereas your normal genotype does make those colors. Does that make sense? So that's called mosaicism, because only some of the patches of cells have that mutation. They accrue it at some point. And some of those chromatin things.
Does it come from like, would they be heterozygote, like normal pigment, and then some cells, gain a mutation where they become homozygous recessive?
It doesn't have to be. So, I mean, for example, there are people with very, very dark skin that have those patches, right? So it can be anything. And it doesn't have to be that you accrue a mutation. It could just come down to gene expression. So remember, we talked about all those ways gene expression can be monitored. So maybe for whatever reason, over that melanin gene, there's heterochromatin, and that gene just doesn't get expressed in that little patch of cells for whatever reason, and so therefore you have no expression of pigment in that region. But for whatever reason, those cells are being affected.
So it could just be a problem in gene expression in itself, not really just.
Doesn't have to be a mutation. It could be, but it also could be due to gene expression.
Okay, thank you.
Okay, so let's look at this family right here. So here's the mom. And then these are her 5 kids, all right? 3 of her kids are albino. So what is the genotype of these albino kids?
AA.
Well, determine, it's case sensitive, so tell me the case. Capital or lowercase?
Oh, lowercase.
Lowercase, little a. So what's another way to say it, like over a phone call instead of AA?
Homozygous recessive.
Homozygous recessive, right? Okay, so if these 3 kids are homozygous recessive, what are these 2 kids? Do we know?
They could be homozygous dominant or heterozygote.
Or heterozygous. So we don't know what they are, right? We just have to kind of say we aren't sure. What about mom? Do we know mom's genotype? Yes, exactly, Elaine. Mom has to be heterozygote. Why does mom have to be heterozygote?
She has to pass on that recessive
So we
gene to the
know what
albino.
dad is, right? Or at least we can know. We can't see dad, so we don't know what dad's phenotype is. What are the possible genotypes for dad? I mean, dad's
He
not
could
this
be homozygous recessive or
good.
heterozygote.
That's right. He could be heterozygous or homozygous recessive. Excellent. Okay. So you can see that once you start to understand the genetics of something like this, you can you could even go back in time and determine what people are. And we're going to talk about pedigrees and how we study genetics in humans in just a bit, and we'll come back to albinism when we do that. All right, now when we're studying genetics, we often want to do what's called a Punnett square to help us understand the relationships and the possible outcomes from a particular mating. So let me just tell you a couple things, and this isn't going to matter too much because your test is not take home this year and it's not short answer, it's going to be all multiple choice, but it's good to get into good practices irregardless. So typically we use a capital letter to represent the dominant allele, a lowercase letter to represent the recessive allele. I suggest you always choose a letter that's easy to distinguish the capital from the lower, so like A. So some of you might have seen like Elaine used P earlier. P is kind of a tricky letter because sometimes it's hard to tell the capital P from the lowercase p. So I like to use a letter where the lowercase just looks completely different, like A or R or G, right? That they don't look alike. S and T and F, oh, it's very difficult to determine the capital from the lowercase. And then another good habit to get into is always put the capital letter first. All right. So we're going to practice making Punnett squares to help us study the outcomes of a possible mating between two different people. All right. Punnett squares are going to help us determine genotype, and then we're going to look at genotypic ratios here, and phenotype and phenotypic ratios. Okay. All right. So let's look at how to set up a Punnett square. First of all, you have to write down the genotypes of the parents. So in this case, we're going to mate a heterozygous dad to a heterozygous mom. Okay? And then you want to make sure you know the possible gametes that parent can make. So you have to kind of go in your head, like, I got to think about meiosis. I got to think about the outcome of meiosis, which is a sperm or an egg, and they're only going to have one of every chromosome, which means they're only going to have one of every gene, right? So a sperm can either get the big D allele on one chromosome, or it can get the other chromosome in a different sperm that's the little d. So it can't have both. It can have big D or little d. Does everybody understand that? Think about meiosis. Okay. And then the same thing goes on with the mom gametes, right? So if this is an individual who's big A, the only possible kinds of sperm he can make are big A. However, if this person is big A, little a, half their sperm are going to be big A, and half their sperm are going to carry the little a allele. All right? Keep in mind, gametes only carry one, right? Somatic cells and zygotes carry two of every allele. All right, now once you've figured out the cross and you've figured out the alleles, you are going to then draw a Punnett square. So a Punnett square looks something like this, where you have empty squares. I always put the dad's gametes across the top and the mom's gametes across the bottom. All right. And then basically you're going to just fill in the square. So you're going to put dad's gametes here, mom's gametes here, and then you're just going to mate them together. So if this sperm fertilizes this egg, this individual, this offspring is going to be homozygous dominant. Did everybody see how that works? So when these two people meet, this is one possible sperm-egg combination for this particular gene, right? If this sperm fertilizes this egg, then that person will have this genotype. Same with this one. And then finally we have that sperm and that egg. Oh, that is a mistake.
Yeah.
It probably, you know how it does that in Word where it capitalizes when you don't want it to? I can't fix it. Well, that's supposed to be little d, and for whatever reason I can't fix it. So you guys know that's supposed to be little d right there, right? Okay.
Yes.
So, little d. Okay, so we have to make sure that we fill that in right, and then obviously I didn't do that right. And I think it's a good idea to write the phenotype under it, because later you might be asked a question like, what percentage of the offspring are dwarf? Well, if you've already written it there, you can easily see, oh, 1 out of 4, 25%, whatever, right? You can see very quickly what the percentages are. Oftentimes you are asked to determine the relative proportions of genotypes and phenotypes. So you might be asked to determine the genotypic ratio, which means how many homozygous dominant, how many heterozygous, how many homozygous recessive. So you just count each of them and put them in a ratio form, or the phenotypic ratio. So there's tall and there's dwarf, how many tall, how many dwarf. All right, so those are just ways you may have to answer those types of questions. It's going to get a little bit more tricky when you do single gene crosses like this. It's fairly simple, but when we get a little bit into double genes and things that don't follow Mendel's patterns, it gets a little bit more tricky than this. Okay, so just keep in mind the law of segregation, how you're never going to get in a gamete 2 of the same letter without it being a mistake, right? So unless there's a mistake, you're not going to get that. Oh, I thought I had some practice problems here. I guess they're coming up. So Mendel started with looking at just one gene at a time, and then he called this a mono hybrid cross, mono meaning a single difference between the 2 plants. So then he started thinking, I wonder if any of these traits are inherited together, right? So he started looking at what's called a dihybrid cross. So he now was looking at 2 different traits at the same time. So let's look at this example that he looked at. He looked at seed texture, so whether the seed was round and smooth looking or wrinkled. And then he looked at seed color. Was it yellow or was it green? All right, so seed texture he was suggesting was determined by gene R, and seed color was determined by gene Y, okay? And he took true breeding ones. So he took ones that were yellow and round, and he mated them to themselves, and they always gave yellow round. And he took ones that were green and wrinkled, and he mated them to himself, so they were always green and wrinkled. So he knew that they must be homozygous in this way, okay? And so then he thought, well, what is going to happen when I mate these together? All right, so there's basically 2 different possible outcomes. It could be that they are inherited together, so that the R and the Y always go together. Okay, he called that dependent or linked assortment. We now know that in order for that to be true, those 2 genes would have to be on the same chromosome, right? Because the whole chromosome goes to one cell. So if they're on the same chromosome, they're much more likely to be inherited together, right? However, if they're on 2 different chromosomes, if chromosome 1 has gene R and chromosome 2 has gene Y, then they don't necessarily have to go to the same cell the same way, all right? And that would be what's called independent assortment, okay? So his hypothesis was, if it was dependent assortment like this, and he didn't know the word chromosome, but they were inherited together, which would mean they were on the same chromosome, the outcome would be no different than what we saw for each gene individually. If it was independent assortment and they were not inherited together and they could each individually be independently inherited, you wouldn't get just that 3-to-1 ratio. You would get a combination of traits such as this, where some would look like the original parents, round and yellow. Some would look like the original parent, green and wrinkled. And then you would get these ones that look not like either of the original parents. So notice you have yellow wrinkled and round green, which are not here in any of the original parental types. So it turned out this is what he saw when he did this with his pea plants, that the genes independently assorted from each other. All right, so this was the original cross he did. He took the homozygous green and wrinkled. He mated it to the homozygous yellow and round. He got all yellow round peas. He mated them with each other, and he got this whole slew of new kinds of offspring. And he did this over and over again, and he found that the ratio was very, very consistent. He found that there was always this phenotypic ratio of 9 of them out of the 16 possible outcomes looked like the original yellow round parent. 1 of them, just 1, looked like the original green wrinkled parent. And then there were these in between ones that had, there were 3 that were smooth but green and 3 that were wrinkled and yellow. So we saw very predictable 9 to 3 to 3 to 1 ratio. Okay? So when he looked at this, he found that there were some that were non-parentals. He called these the non-parental types. And this contradicted the dependent assortment. So this is what led him to think that it was independent assortment. All right, here's some of his numbers that he got maybe calculated, right, that gave him, that led him to another law. So the first law he came up with was the law of segregation. Even though he didn't know chromosomes went through meiosis, he predicted it. And he also predicted the law of independent assortment, and that has to do with how the chromosomes align during metaphase 1 along the metaphase plate, which one the mom or the dad's on the right or the left. And it's completely, every chromosome pair is independent of the neighboring one. Remember how we talked about there are 8 million different combinations? He was predicting that way back in the 1800s. Okay? So just how they align is completely independent of other chromosomes around it. All right, now the tricky part for figuring out these kinds of Punnett squares is figuring out the gametes, right? So this looks like a really confusing picture. Oh my gosh, what in the world, right? So we're going to do it an easier way to figure this out. We're going to use the FOIL method. You probably learned this in math. It stands for first, outside, inside, last. The important thing to remember is you're never going to have 2 of the same letter in a gamete unless something went wrong. So in a normal way, if you think about this working in a normal fashion, you are never going to have 2 A's in a gamete. You're never going to have 2 B's in a gamete. But you will have an A and a B. You will have 1 of each. All right? So make sure when you're done and you figure this all out that your gametes have 1 of each letter. Okay? So let's look at this. If this was our original parent type, heterozygous for A, oops, you guys can't see my cursor, heterozygous for A and heterozygous for B. All right, if we're going to take this, we're going to pair this A with every possible B. So we're going to pair it with the first B, which is big A, big B. Then we're going to pair this A with the other outside B, which is big A, little b. Did everybody see how I did this? Would I ever pair this A with this A? Why not? Why wouldn't I pair that A with that A? That's not how FOIL works. That's right. Because in gametes, these are on the same.
Because in FOIL, it's part of the same. It's part of the same problem or whatever that you're multiplying against. So you wouldn't multiply it against itself.
Right, and in this case, you're not going to inherit 2 A's because they're on the same chromosome. So you're not going to be able to pair them together because those homologous chromosomes separate from each other, right? And so, like Seth said, that would be a bad mutation. The only time you would ever get 2 A's together would be non-disjunction, which is what we talked about with trisomy 21. That would be it. That would be the only time that would happen. And that's a rare instance, really. Okay. So then once you've figured out all the possible A's, all the possible B's that this A could pair with, then you move to this A and you pair it with the inside B and then you pair it with the outside B. So you get little a, big B and little a, little b. And these are your 4 possible gametes. Now, for us, this is basically the max you're ever going to have to find. So the max comes from when you're heterozygous for both genes. you're going to end up with 4 gametes every time. All right, so let's try this one. If you were big A, B, little b, oops, my answers are popping up. If you were big A, little b, what would your possible gametes be? Big A, little b, and little a, little b. Good, 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 good. Okay, and Elaine did it 4 times. If you see what Elaine did, and you can do it 4 times, but really, in reality, half the sperm are big A, little b, and half are little a, little b, and so it's just more work to do it 4 times. But if it makes sense in your brain that you have to do it 4 times, that's totally cool. Okay, and then what about the bottom one? Big A, big B, little b. What are my possible gametes? Big A, big B, and big A, little b, and that's it, right? Excellent. Good job. All right, so that's how we figure out our gametes. Now, that's not to say that every gene is located on a different chromosome. So we are not going to get to this semester. This is one of those things you're going to have to take genetics to learn. Studying genes that are on the same chromosome is super interesting because their inheritance pattern, you would think, would be just really easy. They get inherited together. But what would contradict? So look at this picture on the right. A and B are on the same chromosome in this picture. Why would we sometimes not get big A and big B together? Exactly, because crossing over can happen in between it. So let me just tell you a little story, and this won't take very long. When I was in college, we hadn't sequenced the genome yet. And so there was this desire to try to map our chromosomes. And we would use crossing over as a way of determining what genes were next to other genes. And so genes that were really close together on the chromosome would have a much higher chance of being inherited together because there's much less of a chance that crossing over would happen between them, whereas genes far apart on the same chromosome have a much higher chance of crossing over. So you would do all of these crosses and count the number of ones that were non-parental types, right? And then you would, and then that percentage you could use as basically a frequency to go back to determine what genes were more closely related or closely located than other ones. And so it was like a mathematical challenge, or it was a fun challenge to figure out how these genes were aligned on the chromosome. We don't really probably do that very much anymore because now we just know where the genes are because we sequenced the genome. But for, I mean, probably 40 years, this was the way we were learning where chromosomes or where genes were on the chromosome. Okay, so if we were to do Punnett squares to predict this, the Punnett squares would be bigger, right? You saw that one in the last example that was 16 square Punnett square big, right? So if we go back, let me just show you that one again real quick. Where was it? Right there, right? This is a 16 square Punnett square. So now you can see where we got those gametes from, right? There were 4 from that heterozygous for both genes parent, 4 for that one, and then every combination of those possible gametes mating with each other. All right, so let's do some examples to practice. My suggestion to you to not waste time and not waste energy is only make the Punnett square as big as it needs to be. So that's what I was saying when you don't have to, if it only makes 2 different kinds, then you only need to represent those 2 kinds. You don't need to double it and represent them twice. All right, so this would be a typical 16 square Punnett square. This is only the result of crossing a heterozygote for 2 genes times another heterozygote for 2 genes, all right? That gives you that 9 to 3 to 3 to 1 ratio. All right, so let's do this one. We're going to cross heterozygous for A, homozygous recessive for B, to someone who is heterozygous for A and heterozygous for B. So do my rules, figure out the gametes for each parent first. So figure out the gametes for heterozygous for A, homozygous recessive for B. Okay, we can have big A, little b, or little a, little b. All right, now figure out the other parent.
Big A, big B, little a.
Big A, big B. I got that one. What's the next one? Big
A, little
little b. And then big little a, big B, little a, little b. Right? So my advice here is if you look at what Elaine wrote right here, she's not wrong at all, but I would always put the same letter at the beginning, right? So this one right here that's little a, little b, big A looks funny to me. It just doesn't look right. And I tell you this because when they get complicated, if you don't keep track of what you're doing, it gets a little bit complicated. It gets tricky to follow. Okay, then you're going to build a Punnett square. So how big does your Punnett square need to be? How many columns, how many rows? Is it 4 by 4?
4 by 2?
4 by 2, good. 4 by 2, right? So you would fill in your gametes and then fill in your offspring. And see, you could do a 16 one, but it would be a waste of your time because the bottom 2 rows would be identical to these top 2 rows. So to me, it's just a waste of your time to fill those all in. All right, let's do one more. How about this one? These are unique parents. So the first parent, what are my possible gametes?
Big A, little b, big B.
Nope, big
And
A,
small
little b, little a, little b. Good. What about the other one? Other parent.
Big A, big B, big A, little b.
Good. Okay. So now my Punnett square, what do I need? How many?
2 by 2?
A 2 by 2, that's right. All right, and then you just fill it in and then you answer questions regarding it. Does that make sense? Everybody see that? Excellent. All right. Now, obviously, we can look at 3 genes at a time, right? And this gets very cumbersome because our Punnett square, if we were looking at heterozygous for all 3 times heterozygous for all 3 would give us 64 squares, right? And you can see here, this is actually the Punnett square for skin color. There are actually 3 genes that influence skin color. And so let me go to the next picture here. Oh, I guess I don't have it. So you can see here, this would be someone very, very light skinned. This would be someone with very, very dark skin. And then look at all the options in between, right, that you have. And that's why we have such a diverse range of skin color, is because of the fact that it's controlled by all of these different genes, right? So we wouldn't ask you really to do a 64 square Punnett square. There is an easier way to do it. It's called the fork line method. So basically what you do is you figure out what is the possible for each gene by itself. So we know that tall is completely dominant to dwarf, so we have 3/4 tall to 3/4 dwarf. We know that round is completely dominant to wrinkled, so of the tall ones, 3/4 are going to be round and 3/4 are going to be dwarfed. Of the dwarfed ones, 3/4 are going to be round, 3/4 are going to be wrinkled. Same thing with the yellow, but you have to do it for each and every possible. And then you just multiply out. So 3/4 times 3/4 times 3/4 gives you 27 out of 64 tall, round, and yellow. If you do 1/4 times 1/4 times 1/4, you're going to have 1/64 that are dwarf, wrinkled, and green, and then every combination in between. So that's an easier way to do it than to draw the 64 square Punnett square. Okay, and I probably won't ask you to do that anyway. All right, the next little bit here in this chapter has to do with determining probability and statistics. So now that we know that we can come up with these Punnett squares and we can think about the probability of each individual gene, how do we really use this in a useful way? And people use probability and statistics all the time to help them solve a problem. So for example, a farmer or an animal breeder, they're going to make sure that they get the type of crop or animal that they're looking for, right? So they're not going to just choose random things to breed. They're going to be very careful about what ones they pick because they know they're going to give a higher percentage of the outcome that they're looking for. You might think about this in terms of like genetic counseling. So parents might go and find out what's the probability that I'm going to have a child with this particular disorder because, you know, I have family members that have the disorder and I want to, you know, I want to know my statistics before I go out and I actually have kids. All right. So that's something that's becoming more and more common, I think, is going to a genetic counselor. So probability is basically what's the chance that an event will occur. So how you figure it out is you think, how many times does this event occur, and you divide it by the total number of events. So the easiest to think about is a coin flip, right? What's the probability that I flip a head? Well, head's only on one side, so the number of times that event occurs is 1. The total possible events is 2, so I know that that is half of the time I should get a head, right? So that's a pretty easy example. We could do it in a genetic cross. What's the probability from crossing a heterozygote to a heterozygote that I get an individual that's tall? Well, I know that if I cross those two in my Punnett square, I get 3 tall out of the total 4 possible outcome or offspring. So that's going to give me 3/4 or 75% chance. So it's kind of like using math to do the Punnett square that we had already figured out. All right. Same thing with dwarf. Okay, so obviously this is completely dependent upon whether your sample size is random, how big your sample size is. You have to kind of know, you have to be aware that your sample size can influence you. So for example, let's just say I flipped a coin 10 times and I get 7 heads and 3 tails. Does that mean 70% of the time I get heads? No. It just means my sample size is way too small. And that's totally possible. If you did it 10 times, you totally could get 7 heads and 3 tails. But if you were to do this 1,000 times, you would get 50-50. You would be way, way, way closer to 50-50. So this is why sample size is so important. This is why the vaccination trials are so important, because if you just test it on 1 or 2 people, I mean, it happened with that hydrochloroquine, right? So yeah, 1 person felt better. I think they did the first study with it was 6 people, and like 2 of them felt better after they took this drug, right? Well, that's totally not a big enough sample size, right? And once you go out to the bigger numbers, now people are dying in larger amounts due to the drug, not even due to COVID-19. And so you really have to, oh, did they really? I didn't even know that. But you really have to have a good sample size. And this is why you can't rush the vaccination process, because you have to start with a small sample size to make sure it's not going to kill people, right? And then you have to go a little bit bigger and get all the possible side effects and outcomes, and before you're going to go out to the main public. And so the reason clinical trials and things like that work is because and happen is because we've learned from the past that you can't go too much longer. Okay, I know you guys have lab, because don't you have your lab presentations today? Yeah, so I better stop. So we will start here on Tuesday. So my recommendation to you is read chapter, I think it's chapter 11 over genetics this weekend, because we're going to cover it on Tuesday, and then some of you are taking the test on Thursday, especially if you're taking the test on Thursday. Don't blow off reading it this weekend. You must get to it and read it, because you're going to want to have thought about it before we talk about it so that you can be ready to study for it in those last 2 days that you have. Okay? All right, so study hard this weekend. Write your papers. Finish your vocab project. It should help you study. Finish your genetic disease project. It should help you study. And I'll see you next Tuesday, unless you guys have questions. You have a good weekend too,
I have a
Amy.
quick question.
Okay, go for it.
Are we gonna have like a practice test or anything?
For the final?
Yeah, just I mean, I know that you said like the post-lecture quizzes are good practice tests, which obviously I plan on doing, but I mean, I don't know. Like, it's gonna be sorry, go ahead.
How about this? I'll put it on my to-do list, and I'll try to get to it. How's that? I'm not promising anything.
Well, okay, that's fine. I guess what
Okay.
I'm curious though, is it gonna be more like questions based off of previous tests, practice tests, or is it gonna be whole new questions that we need to in-depth study each chapter all over again?
It is going to basically be like previous practice tests. I will say in general, it's not going to be as hard as my old multiple choice that we used to have in class. So there'll be more
Okay,
big,
perfect.
broader questions than my old tests were. I'm not going to use my
That's fine.
normal.
I was just
Is that basically what I'm saying? Is I'm going to build it new based on our situation and make the questions more straightforward and big picture.
awesome. Last thing is if we, like, when I turn my PowerPoint in, can I share it again? I don't know what's going on with my email, but my email's not working for any of my emails. I don't know if something happened or what, but I can't email anything to anybody right now.
Okay, so what are you asking if you
Once I finish my vocab project, is that okay if I just share that with you?
Yes, that's fine.
Okay, as of right now, it's shared, but I was really curious about. Sorry, thank you.
So you want me to see if I see it? Yeah, give me a second. I'll log in.
Sure. Through my actual email, I guess, app, I'm not sure. It's not letting me email anything. But when I tried sharing it, it said that it was successful, so I don't know.
Okay, let me go to my email real quick. I didn't have it open, so I got to open it. Yeah, I have it. You shared it with me.
Okay, awesome. It's long, so I'm sorry. I didn't know how else to put it.
No, okay, they're all long. This is probably the best way to do it over Google Slide as opposed to an email anyway.
Okay, awesome.
So yeah, they look good. I'm scrolling through them. It looks good. Ooh, I like your climate change.
Okay, great. I liked it too. Thank you.
Okay, anybody else have any questions? Forgot to take attendance again.
Hi, doctor.
Yes, Mohab, do you have a question?
Yes, please. So, doctor, like about my vocab project, like which ones are the ones that I should change, doctor? Like because all 80 or like the examples that I should change, not all 80, right?
No, just the examples were where I think your examples weren't quite specific enough or related to what we were doing.
Sure. So like all 50 examples
related to ours. Yeah, all 50 of them though. Yes. I didn't look at every single one of your 50, but the first 10 I looked at didn't have good examples. So my guess is, the results not have.
Try to.
Does that make sense? They just need to be specific.
Yeah, sure. I'll do that, doctor. And the genetic disease like project is due next week, right?
Next Wednesday. Yep.
Sure. And this is gonna be a paper or?
Yes, you're going to write a paper on your disease, and you're going to have to talk about the molecular biology, the normal gene, protein, the mutant gene and protein, how it differs in function, and then how it affects the cell and how it's inherited.
Sure. Thank you, doctor.
Mm-hmm. Gretchen, did you have a question? No. Juan, did you have another question?
I'm just going to say, just in case, I don't know, something comes up, you said the vocab project wasn't due until next week, right?
Last day is next
week.
Wednesday.
Okay, that tells me. I'm totally going to try and get that done by Saturday, but just in case anything happens, that's why I was asking.
Yep, that's good. The reason I made it Wednesday is because it should help you study for the final, really. I know it's a little bit more work than just studying, but you need to take the test on Thursday anyway, so that's why it's due then. Plus, I have to have time to grade it. Those ones take me a while to grade.
No, that's fine, and it actually refreshes quite a bit, honestly. I didn't think it's going to as much, but it definitely does.
Yeah. That's the whole point of it. I promise there's always a method to my madness to make you study, right?
No, it's working for sure. But like for mine, are my examples okay?
Let me go back and look. I had moved away from it, so let me look.
I'm sorry. You can email me. I'll see if my phone email works, but on my computer, it's just not doing anything.
Okay, an example which activation is when I, yeah, no, that's good. Would be the, yeah. What I'm looking for are real specific examples. So like for anaerobic respiration, you might say, because you said without presence of oxygen, so you might say the cell, instead the cell uses this as its final electron acceptor or does fermentation, right? So that one maybe you could be a little bit more specific on. But that
Okay,
one,
so it's that sulfur, I think. An example is sulfur, right?
yeah,
Okay.
that one's good.
Okay.
Like you said, G1 checkpoint, like that's what I'm looking for, a specific example.
Okay.
Like for your central dogma one, you can give a better example. Like you can say the gene for hemoglobin, right, is made into an RNA for hemoglobin, which is then translated into a protein called hemoglobin, which does this function, right? So you can be more specific for central dogma. Like I would have taken a point off for that one.
So they're kind of hit and miss right now.
Yeah, that looks like, but I mean, you've only had 2 wrong in the ones I've looked at.
I feel like I was as specific as I could possibly be, but some of them I felt like I wasn't sure if I was on the right track, so that's why I wanted to email it first.
I would say use a specific, like I liked how you said an example of dehydrogenases during oxidation of glucose. I mean, you could even say that uses glucose 6-phosphate dehydrogenase or whatever, hexose kinase or whatever. You know, you could actually give an, I guess not hexose kinase, but give an example.
Okay.
Mm-hmm. Does that make sense?
Yes.
So are you telling me you're done
have,
with this, or you're not done with this yet? You're going to share it with me again? Like, should I go
I'll
in and
share it
grade
with
this?
you again.
Okay, I'll wait.
Yeah, I'm totally not done. I have literally about 60% of it done.
Okay.
But that's the next thing I was going to say is could you scroll down to the compare and contrast? So it's really the last 5 slides, I believe.
Okay.
So I'm sorry, I know it's long.
That's okay. Just imagine me, I have to grade all these.
I know.
So I see you're not quite done, like you have vesicle and nothing written yet.
No, there's some that I don't have at all.
Yeah.
Like
so just make sure when you're totally done, you retell me so that I go back and look at it again, especially if you're not going to re-email it to me.
Okay.
Hold on, it's not there yet. It's slow because
no, I know, that's why I felt bad because it loads slow. I was like, I don't know if this is good or not.
So you have to define alpha helix, which you did, define double helix, and then tell me how they're similar. Yeah, that's good. You did good with the alpha helix and double helix. Is that what you wanted me to look at?
Yes, so I don't necessarily need to have those in a paragraph. I can bullet point.
Yes, that's okay. But I would
Awesome.
say, like,
That's what I did with all 4.
With your definitions, like covalent bond, you just wrote equally shared electrons. I would say the interaction between atoms when electrons are equally shared. You know what I mean? Because it's more than just electrons, right? There's atoms involved here. You know, so.
I honestly wrote notes on some of those, so I'm glad you said that. I probably forgot to go back to that.
Right. And then remember your tRNA and your mRNA is not done. But yeah,
Yes.
you're the first one, which is probably the one most people are losing points on, you did a very good job on. Because a lot of people aren't mentioning DNA and protein. They're just, I don't know what they're thinking.
They're just going for shape.
Yeah, I guess.
Okay, I was just making sure, because like for the cilia and microvilli one, I had a ton of information. I didn't know if you needed all of that or not.
I would say I would err on the side of more than less.
Okay, cool. And like I said, I'm not done with everything. I just wanted to make sure I was on the right track. So far, I'm just hearing better examples on the ones that I didn't give specific ones.
Yeah. And like
okay,
for your
cool.
cilia, oh, you do say that one.
For which?
Never mind, you said it. I was looking to see if you said microtubule and microfilaments, but you did. I always look for that. That's like the one we talked about most importantly.
Okay, awesome.
Okay, is that all?
So for tRNA and rRNA, I just need to explain that one is a transfer, one is ribosomal, one helps read while the other one brings amino acids and creates a peptide chain.
Yep.
Like together.
Yep.
Okay.
Yep.
Okay, cool. Sweet. That's everything for me. Sorry, Chet. Oh, he just left, I think.
Yep.
Dang.
Good.
Okay. Thank you so much. I'm sorry about
You're
that. I just wanted to
welcome.
make sure.
You're welcome.
Thank you.
Anytime.
Have a
Just
good
email
weekend.
me. Okay, you too. Bye.